Hi everyone, it's Mr. Stratton. Today I want to give you an introduction to biological molecules, AP Biology topics 1.2 and 1.3. If you look over to the side, you'll see two molecules there. Those are actually the same molecule. They're sort of a stereotypical amino acid, and I can tell that because I see the backbone of the atoms that make them up, nitrogen, carbon, carbon. That's really typical of an amino acid. So what I want to focus on today is actually the arrangement of atoms within molecules, and the way that the structure and their arrangement affects the function of the molecule. Also, I want to get briefly into how we link together monomers to make polymers, because over the next few days we're going to be talking about the four main biological molecules. So those four main biological molecules are carbs, proteins, lipids, nucleic acids. And they say you are what you eat. The atoms and the molecules in your body came from the atoms and the molecules that you consume, that you acquired either through food or through breathing primarily. The main atoms that we find within the human body, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, CHNOPs, well, they're not all in equal amounts, but the four main biological molecules I just mentioned, they actually all contain carbon, hydrogen, and oxygen. Those are the big three. Those are in every molecule. And then nitrogen's only found in proteins and nucleic acids. Lipids have lipids have phosphorus, so like phospholipids. Not all lipids, but phospholipids do. And then nucleic acids have phosphorus as well. And then sulfur is only found in proteins. So what I want you to think about for just a second is, because they're not all the same, how could you use the different arrangement, the different content of atoms to distinguish between different molecules? So, for example, if you needed to distinguish between proteins and nucleic acids, which two atoms would you look to? This is going to be important later because it's how they figured out DNA was actually the molecule of heredity. So, atoms want to fill their outer shell, right? If you remember from chemistry, a full octet, eight electrons in its outer shell is typically what an atom wants. Now, hydrogen and helium, they only have a 1s orbital, so they really only will accept 2 electrons. But carbon, for example, it has 4 outer shell valence electrons, and it needs 4 more. Where does it get those? It forms 4 bonds. Now, hydrogen's only going to form 1 bond because it needs 1 more electron, because it only holds 2. Nitrogen has 5, it needs 3 more, it forms 3 bonds. So you get that typical nitrogen-nitrogen triple bond, which makes nitrogen, N2, extremely tough, extremely non-reactive. It makes up 80% of the atmosphere, and you don't even know it. Oxygen needs 2 more, so it forms 2 bonds or a double bond. Phosphorus needs 3 more, so 3 bonds. And sulfur, just like oxygen, needs 2. So the atoms that have similar structures actually form similar bonds. Now, if you think about oxygen needing 2, carbon needing 4, if you put together a carbon with 2 oxygens, what you get is, well, CO2. This is going to fulfill the octet for each of those atoms. The carbon's going to get 2 pairs of electrons from each oxygen. So you've got a total of 8 electrons there. And then the oxygen is going to, of course, get its additional 2 electrons by sharing with the carbon. Okay, looking at molecules, you actually have a bunch of different ways you can represent these. These are all the same molecule. These are all methane. And if you look on this one particular picture here, you've got this bond, which actually helps to represent the fact that this is a 3-dimensional molecule. That's shown better within the ball and stick and the space filling model, but that's important because these are all spacing themselves out. The one that's dotted is actually moving back into the screen, and so what this means is that because they're off in all three dimensions, X, Y, and Z, you actually have a bunch of different geometries possible when you've got carbon forming bonds. So carbon is the basis of organic chemistry, and because of those geometries that it can form for those 4 bonds, it gives a huge variety of molecules that can be built from carbon. So everywhere within a molecule there's no atom shown, where you just have like a spot, a junction between bonds, there's a carbon there. It's just usually not drawn. So down at the bottom, I've got all those molecules. Everywhere there's a junction with no atom, that's actually a carbon. So all of those molecules at the bottom there are C3H8O. Now, the key idea in AP Biology is structure and function, right? With these molecules, they all have the same chemical formula, but they do not have all the same chemical structure. As a result, they don't have the same chemical function either. In fact, the first two, 1-propanol and isopropanol, those are both alcohols, and they are liquids at room temperature, whereas ethyl methyl ether is a volatile gas. It's extremely explosive, and while the 2 propanols there are both flammable, they're not explosive like ethyl methyl ether, and they're also not a gas. So why is that? It's because the way the electrons are shared amongst the atoms within the molecule. So in the case of 1-propanol and isopropanol, they've got these OH groups. These OH groups here, what's happening is the oxygen, being more electronegative, is sucking the electrons from the hydrogen, meaning that this is slightly positive. And so because of those slightly positive hydrogens, those molecules are actually going to dissolve in water. Over here, this is an ether group, and the electrons from the oxygen are being pulled in. They're being pulled in towards the oxygen from both sides. The result is there's no one polar place on this molecule. There's no one positive area. And so the result is it really doesn't dissolve well. It doesn't like to stick to itself. And so it doesn't. So they don't hang out together. They make a gas at room temperature. So those little groups are called functional groups because they give the molecule function. So there's actually several important ones that we need to know in AP biology. The first one, the hydroxyl group, that's what we were just looking at. So that OH there makes alcohols. It's also found in sugars. It's one of the most common. It's everywhere in biology. And again, it's making the molecule polar because that oxygen's more electronegative and it's pulling those electrons in. Okay, methyl group, that's going to come up more later on when we talk about modifications of DNA. It's a non-polar group, it's just carbon and hydrogen, and they're very similar in their electronegativities. Okay, we're going to ignore the carbonyl group and move on to carboxyl. So the carboxyl group, what you've got going on there is you've got a double bonded carbon, and then you've got basically a hydroxyl group on there as well. But altogether, it's called a carboxyl group. And with the electrons from these oxygens, they're sucking the electrons in. The result is, again, this hydrogen becomes so positively charged that it actually, in a lot of cases, will completely lose its electron to the oxygen. And when that happens, this is a hydrogen atom. It's just a proton with an electron. And when it loses that, what do you got? That's just a proton. And so that is actually what makes acids, is just naked protons. And those protons can go off and react with everything. And those reactions are what cause the typical reactions of acids. So, okay, we can come back to that in a second there. Amino group, so this is going to be actually a basic group. So that's a nitrogen with 2 hydrogens, but it also has 2 electrons that are just out there as a lone pair is what it's called. Those 2 electrons, well, they're going to attract in protons. They're negatively charged. They're going to attract in a positive charge. And when they attract that in, that's the definition of a base. So this molecule is actually basic. Phosphate, well, that looks pretty complicated right there. It's mainly found, if you think about DNA, the backbone of DNA has phosphate groups. It's also what the little molecules we chop off on ATP. We store and release energy from the reactions of ATP. Biologists are lazy when it comes to chemistry. In fact, I don't want to draw that phosphate group. Pretty much, you're going to see phosphate group, and it's going to look like this. This is the biology way of drawing phosphate groups. Okay, and then sulfhydryl groups, it's actually similar in function to a hydroxyl group. You've got S, which has the same basic electron configuration as oxygen, and the hydrogen out there, and so it's polar. They also like to bond to each other, so you get what are called little disulfide bridges within proteins. Okay, so looking at this molecule, what 2 main functional groups do you see? And this is an amino acid. Think about its name. On your paper, go ahead and circle those groups and name them. And also, there's that NCC backbone I was talking about. Okay, monomers build polymers. So what's going on here is you've got monomers, the individual little pieces, which are like Legos, being used to build polymers, which are like a Lego spaceship, basically. And the way you put those together is through a reaction called dehydration synthesis. D meaning removal, hydro meaning water, so you're removing water from the molecule, and then actually breaking them down is called hydrolysis, lysis being breaking. Right? You think about Lysol. What does it do? It breaks all the bacteria on your counter. So hydrolysis is going to be used to break apart the molecules using water. So what does that look like? Well, when you've got dehydration synthesis, if you take the water out, you see this, you've got the OH and the H here. So those are going to combine together to form water. That's going to leave the molecule. It's going to dehydrate, and then you're going to form this ester linkage between these two molecules. Same thing down here. Hydrolysis is the exact reverse of that reaction. Put the water in, and breaking apart this ester linkage, you're going to wind up with an OH, the hydroxyl group, and the H on the other side right there. So what I want you guys to do is taking a look at this reaction here. So this is showing glucose reacting to form maltose. So 2 glucose molecules making 1 maltose molecule. So what type of reaction is it? What functional groups are involved in that reaction? So your reflective practice for this is go ahead and create a table or model to help you remember which elements are found in which macromolecules. If the 2 molecules below have the same chemical formulas but completely different properties, explain why. So go back if you need to, look at the video, and it'll help explain that. Create flashcards of the 6 most important functional groups. So you need to be able to recognize them. Sorry, that's just memorization. That's all it is. And then identify and summarize the reaction that could be used to link the 2 amino acids below, and summarize how that dipeptide formed could be broken down again. So you're looking at those reactions there. Okay, so looking back at this then, we talked about structure and arrangement of atoms to make molecules and their properties, and we talked about the linkage between monomers to build polymers. So, thank you for watching.